it's a great honor to give a talk at Canadian Pain Society. So given the time difference, I decided to pre-record my talk. Please understand it, but I will try to be available after the talk for Q&A. In this talk, I will introduce one study that aimed to examine dynamic reconfiguration of functional brain networks during sustained pain. So pain is a dynamic experience. It changes with time. And this plot, which is from Balicki 2010 paper, shows how the continuous pain ratings evolves throughout a session. And even with the same stimulus intensity, the pain ratings vary across trials and stimuli. Also, those who reported these ratings actually were patients with chronic back pain. And you can see in this blue line, their spontaneous pain also changes with time and external stimulus input. In addition, these data are from the current study that I'm introducing to you, where we delivered the capsaicin liquid onto a participant's tongue. And as you can see, the pain ratings changes over time, and the ratings are different across participants. So this dynamic nature of pain experience should involve dynamic brain processes. And in addition, it is now well known that pain involves distributed brain networks. So among many relevant references, I really enjoyed this recent paper that proposes the distributed nociceptive system in the brain. Particularly, it suggests that pain processing has a degenerate nature of mechanisms. Therefore, many researchers aim to model pain processes in the brain as dynamic networks and highlighted the importance of studying the dynamic pain connectome, the term coined by Aaron QC and Karen Davis. But still, we do not have the detailed characterization of how the dynamic interactions among multiple brain networks change across the different time phases of pain, that is initiation, peak, and remission. In this study, we aim to model pain as a dynamic reconfiguration of functional brain networks over time, and we are going to model the sustained pain induced by orofacial capsaicin administration. This is the same task paradigm from a recent publication in Nature Medicine. The task design was quite simple. So we collected continuous ratings for the control and the capsaicin runs. Each run lasted 20 minutes long, and the order of the runs was counterbalanced. And this plot shows the continuous ratings over 20 minutes. The red line is for the capsaicin condition, and the blue line is for the control condition. As you can see, the rating plot shows the initiation and peak and the slow remission of pain. And you defined the first 6 minutes as early, and the next 8 minutes as middle, and the last 6 minutes as a late period of pain. Note that we use the avoidance rating here, which asks how much you want to avoid this experience in the future. It was because this experiment also involved other types of aversive non-painful stimuli that actually are not included in this study. And we wanted to use one single rating scale for all different types of stimulus conditions. So using fMRI data in the native brain spaces, we calculated the voxel level connectivity matrices for non-overlapping 2-minute moving windows. So we constructed voxel level functional connectivity matrices in the native space and thresholded the matrices based on the network density 5%. We chose this density level that maximized the differences in a few network level metrics like global efficiency or transitivity between the capsaicin versus control conditions. Then we used the multi-layer community detection method that Mucha has developed before, and then calculated the voxel level module allegiance values, where if 2 voxels were included in the same community, we assigned the number 1, whereas 2 voxels that were in different communities got 0 value. Therefore, this module allegiance matrix actually has a similar interpretation with functional connectivity, but it is based on multi-layer community detection method. And we can then use the module allegiance values for consensus community detection or predictive modeling. We tested the module allegiance based predictive models on one independent test data of n equals 74. The first research question was, how does the brain community structure dynamically change during sustained pain compared to the resting condition? So these maps show the consensus community structures for the capsaicin versus control conditions. It can seem complicated, but I will highlight some interesting differences between these conditions. First, I want to direct your attention to these two areas, so dorsolateral prefrontal cortex and thalamus. You can see the size of the red community became smaller or absent in the control condition compared to the capsaicin condition. We can visualize them using this river plot. Here, the size of the boxes represent the number of voxels. As you can see, the biggest change were found in this community 2, which is the somatomotor dominant community. And this plot shows the proportions of different large-scale networks within the community 2. So you can see the colors of the large-scale networks. The pie chart here shows the common voxels across the capsaicin and control conditions and their large-scale network membership. And the pie chart here shows the voxels unique in the capsaicin condition. First, you will notice the size of the pie chart is much larger in the capsaicin condition compared to the control condition. And the biggest differences between these two conditions were found in the frontoparietal network. Here we are showing the community 2 separately, and the voxels in red are included in the community 2 only during the capsaicin condition. So they included the DLPFC, IPS, and basal ganglia, and thalamus. Thus we can say these regions communicated with the somatomotor dominant community only during pain. Otherwise, during resting, they don't talk to somatomotor network that much. If you see this map, during rest, the DLPFC and basal ganglia is in the default mode network dominant community, whereas the thalamus is in the cerebellum dominant community. And this map shows the large scale brain networks, and the DLPFC and IPS they changed their membership during pain or within the frontoparietal network. So in sum, during the capsaicin condition compared to the control condition, the somatomotor network dominant community was emerged by incorporating some regions within the frontoparietal network, thalamus, and basal ganglia. That is actually consistent with the concept pain supersystem first proposed by Zhang et al. 2019. The supersystem integrates multiple systems to form one large functional network community for pain processing. And the second research question was, how does the brain community structure dynamically change over the course of sustained pain from its initiation to the remission? So to answer this question, we obtained the consensus communities for the early, middle, and late periods of pain and compare them. So I want you to focus on these two parts, and they can be visualized with the river plot again. So there were three interesting observations related to these two parts. The first one was the somatomotor dominant community became smaller, consistent with the previous finding, while the frontoparietal dominant community got larger. And lastly, the limbic dominant community also got smaller. So let's go over each observation. The first one was the somatomotor dominant community became smaller. So this brain map shows the voxels that were included in the somatomotor dominant community during the early period of pain. So here E stands for early and L stands for late, and changed their membership in the late period of pain. And as you can see here, the most of the voxels colored as cyan, that means the frontoparietal network dominant community, community 6. So we can say for early to late periods, a large number of voxels moved their membership from the somatomotor to the frontoparietal network dominant community. And second, the frontoparietal network dominant community formed a large community during the late period of pain. And this brain map shows the voxels. This brain map shows the voxels included in the community 6 during the late period. And as you can see, the frontoparietal regions such as DLPFC, for example, if you can see here, were dissociated from the somatomotor dominant community in the late period, and they formed their own community. And third, this brain map shows the voxels that were included in the limbic dominant community during the early period of pain, and their membership changes in the late period. So during the early period, the brainstem, this brainstem, was connected to the limbic brain regions, including here hippocampus, amygdala, parahippocampal gyrus, and temporal pole, may be related to the spino-parabronchial amygdaloid pathway. But in the late period, the brainstem became connected to the cerebellum dominant community. The third research question was whether we can develop predictive models of sustained pain based on the patterns of dynamic brain network changes. So to answer this question, we first projected the group-level parcellation atlas onto the native space of each participant. Here we used the Schaefer parcellation and added some additional regions including brainstem, cerebellum, and subcortex regions by combining multiple available atlases. Then we calculated region-level module allegiance by averaging voxel-level allegiance values over the regions, and based on time-bin region-level module allegiance, we developed predictive models for the classification of capsaicin versus control and also for the prediction of pain ratings. So this shows the group average module allegiance values for the capsaicin and the control conditions. And they look very similar, but if you trained on SVM support vector machine classifier, it can detect small differences and build a model. I'll visualize and interpret the model in the next slide, but before that, here is the cross-validated two-choice classification accuracy for the capsaicin versus control conditions, which shows near perfect classification. And this is independent test results, which show again significant classification performance, 81%. So now I'm showing the thresholded model to make the interpretation easier based on bootstrap test. Thus, these are the features that make reliable contributions to the classification. And these are the top 50 stable connections. So one interesting observation was the strong negative connections between brain regions within the somatomotor network, which we couldn't really observe with the consensus community detection method. And if we take a detailed look at the results along with the sensory homunculus, we can observe the segregation between the dorsal versus ventral, that is tongue area, S1 cortex region, for the capsaicin versus control. And this tongue S1 area was connected to insula, including deep insula. And if we calculate the weighted degree centrality using the sum of weights separately for positive and negative weights, this tongue S1 area appears to be the top region that shows the largest sum of both positive and negative weights. And using this as a seed, what we can do is that going back to the voxel level module allegiance map, and we can calculate the seed-based allegiance map, which is basically similar to seed-based connectivity, and that provides fine-grained community structure for capsaicin versus control. And this map is one sample t-test result showing the tongue S1 region showed disconnection with other somatomotor cortex regions while showing strong connection with the DLPFC, basal ganglia, and thalamus. So this analysis provides a more detailed description of the network reconfiguration patterns than the previous consensus community analysis, which only provided the patterns of the whole somatomotor dominant community. And this really shows what is happening inside of the somatomotor network. And this result suggests that the tongue area showed intra-network segregation and also inter-network integration, including connections with frontoparietal and basal ganglia and thalamus regions. And the last result is a module allegiance-based prediction of pain ratings. And this is the principal component regression model of pain ratings. And these show that the cross-validated prediction performance and also the independent test results, both tests show significant performance. And this shows the thresholding model based on bootstrap tests again. And these are the top 50 stable connections. This pattern shows strong negative prediction weights in the cerebellum, as you can see here. And this means the cerebellar connections were predictive of pain relief. And this cerebellum region appeared to show the largest sum of negative predictive weights. But to which regions were the cerebellum connected? To answer that question, we did the same seed-based module allegiance analysis, and the t-test results show that the negative predictive weights with the cerebellum were found in the anterior cingulate cortex, DLPFC, and basal ganglia, and some brainstem areas, and insula. And this suggests the important roles of the cerebellum and its connection to the frontoparietal ventral attention networks and the subcortical regions in pain relief. In sum, there were three main findings for the comparisons between the capsaicin versus control conditions. And first, during pain, a pain supersystem centered around the somatomotor network was emerged. And the pain supersystem incorporated the frontoparietal and thalamus and basal ganglia regions. And second, during pain, there was a segregation between the dorsal versus ventral, that is tongue S1 area, while the tongue S1 area showed integration with basal ganglia, thalamus, and DLPFC. Also, there were three main findings for the early versus late periods of pain. First, in the early period, the brainstem was connected to the limbic brain regions. And second, in the late period, the frontoparietal regions were dissociated from the somatomotor dominant community, forming their own community. And third, the cerebellar connections to the frontoparietal and subcortical areas were important for pain relief. To conclude, the current study provides detailed characterization of dynamic network reconfiguration in response to sustained pain and provides new insights into the mechanistic understanding of the network level pain processing in the brain, and also provides potential neuroimaging biomarkers for sustained pain. With that, I want to thank the funding sources and also to Choong-Wan Lab and here Jae-Jung Lee. He did most of the work. Thank you for your attention.